The overall goal of this procedure is to generate human nasal cell spheroids, which can ultimately be used as an assay for functional CFTR studies.
This method can help answer key questions in the cystic fibrosis field, such as assessing an individual subject's response to CFTR-altering drugs in an ex vivo model system.
The main advantage of this technique is that a large number of spheroids are generated in a relatively fast time frame from a small nasal cell sample, allowing for extensive testing. Demonstrating the procedure will be Lauren Strecker and Jessica Moncivaiz, technicians from our laboratory.
After processing and expanding human nasal epithelial or HNE cells on feeder fibroblasts according to the text protocol, bring previously prepared expansion medium, differentiation medium, 0.1% trypsin EDTA solution, and sterile PBS to room temperature over 30 minutes. Then use 70% ethanol to clean the containers and transfer them to a clean biosafety cabinet. In the biosafety cabinet, use a Pasteur pipette attached to a vacuum line to aspirate and discard the medium from HNE culture. Then, with a clean serological pipette, use 5 milliliters of PBS to wash the dish by adding, swirling, and aspirating the buffer. Add 5 milliliters of 0.1% trypsin EDTA solution to the culture, and return it to the incubator at 37 degrees Celsius and 5% carbon dioxide for 5 minutes. Then, under an inverted microscope at 4 to 10x, check that the cells detach from the dish, become round, and float. Gently tap the side of the culture dish to dislodge the cells, and if necessary, re-incubate the dish for an additional 5 minutes.